Thank you, Rich. Thank you, Bill. It's really a pleasure to be here at this meeting. I feel it's one of these meetings where it's so diverse and we are coming all from a different direction. It's more about explaining our motivation for which problem we want to tackle, but really also to discuss the approach we take. Maybe that's more important than really the details of the talk I will present. So combining theory with biology, that's really what our goal is. And so thank you for having me here. Our interest is in developmental biology, and it is true. We are studying rhythms, timing, oscillations. We want to understand how the temporal organization is connected to the spatial organization of developing embryos. But I thought before I show you these incredible embryonic rhythms, we have seen some of them in the previous talk, that was really beautiful. I wanted to show you a more general motivation, and I share with you the most spectacular example of emergence. We had a wonderful discussion yesterday about emergence at your poster. It was great. This is an example I was made aware of at the Gordon Conference for Chemical Oscillators. It's an amazing meeting. You should really look it up. I went there not really knowing what to expect, and for 5 days people would discuss BZ, BZ, BZ. BZ is Belousov-Zhabotinsky reaction. Many of you know that reaction. You should look it up on Google. It's an amazing chemical reaction that is oscillatory under certain conditions. The redox state can oscillate. And the whole community at this meeting discusses the dynamics of that system. And so after my talk, Ken Showalter comes to me and says, look at my cells. Look at what my cells are doing. So here you see Ken's cells. These are plastic beads that they call cells. They are porous. They're loaded with the chemistry. BZ chemistry, of course. They have more chemistry that oscillates, but that's really the premier. And nothing happens until you have 89 of those. He says nothing happens with 88. Okay, you should look up the paper. It's a very sharp transition. Okay, just imagine one more beat comes in. I love the idea of collectives. One more person comes into this room, number 89. Look what happens. I hope the movie plays, but it will play. Bam, did you see this traveling wave? Very similar to the wave we just saw in the previous talk in a way. Except that this is Belousov-Zhabotinsky. It's a reaction diffusion system, and the redox potential spreads as an oscillatory wave through this bead collection only if you have 89. You take one bead away, everyone stops. Okay, I thought what Ken showed me was quite amazing in a way. On the other hand, we know that quorum sensing, and that's the title of this paper, quorum sensing is very pervasive, not just in biology, across all different contexts. And this is what we see here. It's a quorum sensing system that at 89 undergoes a transition. Everyone becomes oscillatory. And in addition, you make spatio-temporal wave patterns. I think that's really an amazing example. It made me think a lot during these 5 days. By the way, it's a meeting with a lot of theoreticians, and that was one of the motivations to go. It was a really good mix between theory and chemists, engineers, mathematicians. It was a terrific meeting. And I think what I took from that was a couple of things. First of all, of course, collective rhythms that I study in biology are much more pervasive. Chemistry shows them. They emerge at all scales. But also, maybe more nuanced, is the notion of function. Here I show you the beat at which this wave started. We could call it a pacemaker. And at least in my field in biology, we might look now for features of this pacemaker. We have very sophisticated methods to take them apart. We might look for features at one level or the other, the RNA, the protein, something of this beat that makes it the pacemaker. And it's clear in this context that this is not a pacemaker. It has become a pacemaker. Anyone in this 89 could be a pacemaker, depending on the geometry. And in any case, it only happens if you're 89. So I just thought, think about it. These chemists know everything about this reaction. They actually put it in. They load the beads with the chemistry they know. And still the question is, why is this one becoming the pacemaker and not the neighboring one? We got the question about where do the waves emerge? It's a key question. And here it's a system that at least you don't have to worry about not knowing the components. And still they meet since the 70s, and they still meet for the whole week to discuss the emergence of that pattern. And in this system, they know quite a bit about it. But it made me think because, of course, in biology, we face a much more challenging problem that we don't know the chemistry and we don't know all the components. And that experience just reinforced my view that the only way to proceed with the complexity of biology is to integrate theory and to abstract. Because even if we knew all the components, this shows us that it remains a challenge to really understand the emerging dynamics of the system. So for us in biology, as I said, it's much more complex. I'll show you only two examples of rhythms that we study. One of them is a clock that is ticking in all vertebrate embryos. And I'm going to assume that this is not known to most of you. In our field, we call it the somite segmentation clock. And on the left, you see a mouse embryo, and the color shows you the onset of this clock. It's a signaling pathway that I will describe in a second. But for the moment, it's sufficient that you see the dynamics of it. The cyan color is a new mesoderm, it's a new cell layer that forms the mesoderm. And you see that over time, the first waves emerge. It's the onset, the first time in development, that a vertebrate embryo shows traveling waves that go through the body from the posterior to the head. On the right side from you, you see a fish embryo at a very early stage in development. I show you a calcium sensor that we have seen many times in the neuronal setting. These are not neurons, these are cells before gastrulation. These are very early cells. And have a look at their dynamics. You see calcium dynamics in these early embryonic cells. Each cell is spiking every 2 minutes. And then the longer you see, things become collective again. And you see that local clusters emerge. It seems a bit random at first, but the longer you let it go, you see that the waves become more and more regular and more directed. And we know that the directionality matters in this case. This is again the body axis that forms in this very moment after the cells become collective. And the point here is that rhythms are pervasive. We see them at all scales, also in biology. On the left side or on your right side, it's 2 minutes. In the mouse, it's every 2 hours. We see it at very different scales. And yet the questions are, of course, very similar. Similar to Belousov-Zhabotinsky. What is the origin of these patterns? What is the nature of the wave in the first place? That can be very different. And in fact, in these two examples, it is very different. But of course, in biology, we have this additional key question about function. And this is what we spend a lot of time thinking about. How would you use such an oscillatory system to encode not just temporal information, but also spatial information? Can you use an array of oscillators to encode space? And these ideas are by no way new. These ideas have been discussed at meetings that I think we are trying to revive with a meeting like here. These are the very famous Cerbelloni meetings. Some of you might know about them. These were initiated by Waddington, and they were happening in the 60s and the 70s. Every 2 or 3 years, a group of theoreticians, mathematicians, biologists, linguists, philosophers would meet to discuss questions that I feel very related to what we discuss here. They would discuss, for instance, proposed by Brian Goodwin, how an array of oscillators would encode positional information in the embryo. And it's a remarkable paper. You can see how long it is. 60 pages of theoretical paper, and you should really read the beginning, how thoughtful he posed the problem. And the ideas that were posed there, I think, is what we're coming back now. And what was very clear back then is that we need a process view, a dynamical systems view on these systems in order to advance. And I think that's exactly what we try to propose now. So the system we study the most to address this question of dynamical systems and the role of oscillations is the segmentation clock. As I said, I assume most of you are not familiar with this clock system. You see here a mouse embryo after 9 days of development. And these somites will form your vertebra, the vertebral column. You can see them already stained here. This embryo has now 10 to 12 somites. And then the somites will differentiate into the vertebra. They will also form your muscles and other tissue. What is particular about somites is that they form periodically in all vertebrates. In mice, they form every 2 hours. And so they would form in this part of the embryo that we call the pre-somitic mesoderm. It's an undifferentiated mass of cells. And if you wait 2 more hours, the next somite pair will form. And then the next and the next. So this is a rhythmic morphogenetic event that happens in all vertebrates. In humans, it takes 6 hours. In zebrafish, it takes 30 minutes. In medaka fish, 80 minutes. Every species has their own rhythm of somite formation. And underlying that activity is this clock that I already showed in the previous slide. Here now I show you a bit later stage, and what you will see when I play the movie, that this particular pathway that is the notch signaling, excuse me, the notch signaling pathway. The notch signaling pathway is periodic in the sense that its activity is peaking every 2 hours. And then again, you see traveling waves that sweep through the body axis from the tip of the embryo towards the anterior of the embryo. And so what is interesting is that when these waves were discovered is that the frequency matches pretty much, I should say, the formation of the somites. It's 2 hours and 2 hours. This structure has about 20,000 cells. And I will not go into much detail of the molecular underpinning. These are, at least in the mouse, these are 3 big machineries: Wnt, notch, FGF pathway, and they all activate about 100 gene targets. And these gene targets show oscillatory transcriptional activity. But what is really particular is if you go in and measure the dynamics of these oscillations. So if you measure the oscillations carefully, you will see that at the tip of the embryo, so where the wave starts, the oscillations are slightly faster than towards the anterior of the embryo, where differentiation happens. So for us, it's interesting that where cells are most stem, they are slightly faster, and then the oscillations slow down, slow down, slow down, and then you differentiate. Of course, this is just an observation. There's no causality in that statement, but it's an interesting observation that in all systems we study, you see this gradual slowing down of oscillations. And so this produces what we call a period gradient. So in this system, along the axis, you have a graded period. So the fastest is at the tip, and then you slow down. And because of that period gradient, we see waves. So these waves might be quite different from what you intuitively think these are. You might think of a trigger wave, one cell activating the next and the next and the next. This is not directly happening here. This we consider phase waves. Each cell we consider an oscillator, and there are beautiful experiments in zebrafish done by Andy Oates showing that if you take a single cell in a dish, these individual cells will show the oscillations and will show the slowing down. Okay, so these 20,000 cells are oscillators. Now you couple them, but still fundamentally these are phase waves that traverse the tissue because of the period gradient creating a phase lag between these oscillatory units. Okay, it's quite different than a trigger wave. And you can test that by, for instance, putting a barrier inside this wave trajectory. If you put a physical barrier, something unexpected happens. I always show that to the students coming in. If you put a physical barrier in, what happens to the wave? Most students say it will stop, of course. But this phase wave will just continue on the other side, because on the other side, cell will continue the oscillation too. What will happen over time is that then if you block communication, these waves become desynchronized. So coupling is important. Fundamentally, these are phase waves. And this is what we wanted to focus on more than all the molecular important details that we also have to understand. And so today I will rather have a high-level view on this dynamics and not showing much of the molecular details. Okay, so this is the phenomenon. And one thing we did in the lab many years ago now, when I actually started at EMBL, we wondered how much can we simplify the system from 3D, from geometry, to 2D. So we take the stem cells out of the context. They are sitting at the tip, as I told you, and we put them on a cover glass that is coated with fibronectin. And now you see a primary culture, you see the cells spreading over the surface and substrate that we provide. And you see a primary culture that doesn't look very spectacular. Many of you will do primary cultures. It's a quasi-monolayer in some parts of this 2D culture. Okay, what about the dynamics? I show you again the signaling reporter that reports notch signaling. And if I play the movie, you will see that not only you see rhythmic activity, but over time, you see waves again. You see waves that sweep from the inside to the outside in this very artificial two-dimensional context. It has really not much in common with the embryo. Everything about it is artificial. Yet the cells reorganize their activity and they produce these coherent wave patterns. And we showed that even in this in vitro setup, the location at which the wave stops is also the location where you form a segment, even in this simplified system. It's really remarkable to us that the oscillations are maintained, but not only qualitatively, also if we measure them very carefully in using chymographs, we see, this is a bit dark, I'm sorry for that, but you can see maybe on the right side a chymograph that we make. Space on the y, x is time, and then you see this very rhythmic activity even in the 2D assay, and the timing is preserved to the 3D in vivo context. We can use this simplified assay to also quantify not only the intensity, but for instance, do a wavelet analysis pixel by pixel, and that has been very useful to become even more quantitative. And on the right, what you see is a representation of the oscillation phase on the top right. So color in that movie represents phase of the oscillator, peak and trough, blue and red. So that's a very drastic step. There is no intensity information anymore in that movie on the top right. And then, of course, we can calculate the period of these oscillators. And again, we see that the period gradient re-emerges also in vitro in 2D. The center is always faster, 135, 140 minutes, and then you have about 20% slowing down to the periphery of this field of oscillators. And that was remarkable, that this very artificial system shows us this coherent re-emergence in a way of phase waves. Now, we use this in vitro assays quite extensively over the years. I show these topics because some of you might be interested in the topic of self-organization. We had some discussion on that. So, for instance, what happens if you randomize these cells, randomize their starting conditions, and put them back into contact, then they will self-organize into small miniature versions of the structure I showed you before. So the waves reappear, the period gradient reappears self-organized in the dish. I will not talk about that today, but I show it here so that we can maybe discuss later over coffee. We also investigated the rules of coupling in this system and found that it's a highly non-reciprocal interaction. Also a topic that we could discuss, and we keep it for lunch or dinner. Today I thought I wanted to share more about really the approach, how to tackle the oscillations in a more fundamental way. Essentially, how can we control these oscillations so that we can go from observations and correlations to really ask questions about function? Because the field, I think, has moved on dramatically in terms of visualizing these oscillations. But the key question is always the same. What is the function of the rhythm? And how could you change rhythm and only rhythm? How could I move from 2 hours to 3 hours and ask what is the consequence for a developing organism? And this is where the input of theory was absolutely essential. This is also what I wanted to show and share with you today. We made use of a theory, an entrainment theory, a synchronization theory that might be familiar to many of you. I think it is fair to say that these Arnold tongues that I'm going to explain to you are usually not taught in developmental biology, and that's a mistake. I think we should all learn about Arnold tongues and bifurcations, and that's why we are here. Also because they're very intuitive. What Arnold and colleagues, it's a whole field, describes is the synchronization between multiple oscillators. And this Arnold tongues, this gray area, is a parameter regime where two oscillators would be coupled. They would be coupled, for instance, in a 1-1 relationship. You would have two oscillators and they would be in the same frequency. You could also get higher order coupling and more interesting couplings. So those of you into music might be familiar with that. You could have 2-to-1, 3/2, and so on. Today we focus on 1-to-1 coupling. And what this theory tells us is that, at least in this representation, you have to tune two parameters to control the synchronization between these oscillators. On the y-axis, you see epsilon. Epsilon is the coupling strength between these two oscillators. On the x-axis, you see the period mismatch between the two oscillators. We call it detuning. Okay, and this is where the Arnold tongue gets its shape. So you see now on the x-axis the detuning. I'm not sure if that is working. Can you try if this is working? Then I can. Yeah, it's not working. Then I stay here, no problem. So it's all fine. So on the x-axis is the detuning, and so here would be omega 0, the natural frequency of oscillator number 1, let's say. And now you come in with a second oscillator that has a slightly different frequency, okay? And that's the the frequency, we call it the driver or the force, the entrainment force. And that is slightly different. And then depending on this detuning, on the mismatch, you can calculate how much force you have to use to land inside the parameter regime, inside the tongue. Okay? So the more you move away from omega 0, the more epsilon you need to be up here in the corner of this tongue. And of course, this is a finite region. If you move too far away, you cannot entrain, you cannot couple. And I think this is very intuitive also to developmental biologists. The more similar you are in frequency, the easier it is to be coupled. And in fact, very little coupling force is sufficient to entrain and to couple. And that is being exploited in many areas, not in biology, but in engineering and in other parts. And that's the rationale. So if we could control the system with a second oscillator, we should be able to control the rhythm of a complex embryonic system. Okay, so that was the idea. Now it's a few years back. The key point here is, again, to highlight that if you talk to physicists, that should work. Any oscillator has to be, you can train any oscillator as long as you match this parameter regime. Okay, so we definitely have to try. The second thing that I thought was very attractive, and again, we had a discussion on that yesterday, I think, to me at least, it was one way to think about targeting rhythm directly. See, targeting at the mesoscale, and not trying, for instance, to change some gene products or some players, which is also important and is being done. We wanted to come in, and that's why I called it top-down, into the system using dynamical systems properties. And I'm saying that because for many of the pathways, we don't have enough knowledge about the players. So we are far away from Belousov-Jabotinsky. What we do have is the rhythm. So we thought, let's try this top-down strategy using physics as a guiding principle. Okay, and that challenge was taken up by Ina Sonnen. She was a postdoc at the time. At EMBL, we have an interdisciplinary program. I'm sure you have that here too. She joined two labs, our lab thinking about rhythms, and a second lab thinking about microfluidics and bubble traps. And that was very important because it took her several years to set up this microfluidic platform that you see here on the top, on which we grow our embryos or pieces of that. So you see them here growing. She included some pillars to keep them in place. And that's a fluidic chip for which, of course, now you can control the flow. And now you see that periodically, in this case, we would pump in a dye, but the goal was to periodically come in with a stimulus, an activator or an inhibitor of the notch pathway, of the FGF pathway or the Wnt pathway. The beauty of that is it really doesn't matter which one of those we use. We used many inhibitors, activators. The key is that this activator comes in periodically, in, out, in, out. So that is the difference. In the field, we have many activators and small molecules we can use. And usually people do dose titration, of course. Here, it's all about the rhythm of the exposure to this drug, to the embryo. So here I show you one of her first successful results. I still really much like to look at this old slide by now. On the top, you see 12 embryos. They all are in their own rhythm. They are not communicating. Each embryo is in its own rhythm. They're not synchronized. And you can see that even though they're all a 2-hour rhythm, they are not in sync. The bottom one are 11 embryos. And now periodically at this gray bar, Ina would flush in a small compound called DAPT, low concentration, in, out, in, out. And you see that after a few hours, these embryos get in sync. Of course, it's noisy. This is not perfect. But that was the moment for us, our Horeca moment. It was really an amazing time to get a sense that we start to be in control of the embryonic timing system from the top with a very simple trick, if you think about it. It still took years, and that is the detail of that. But as an approach, you see how simple in a way it is in terms of assumptions that we make. Paul in the lab was then following up by Ina and asked a subsequent question. So Ina did what we would do. She chose a period close to the natural period, because that's where the Arnold tongue is pointed and where you have to have very little coupling strength to be successful. So if you want to be successful, you start at the natural period, of course. Paul then in the lab tried to move away from the natural period and asked, can we slow down the embryonic system? Can we speed it up? So he spent his whole PhD on entraining many samples. And here you see some of the results. On the x-axis, you see the rhythm of the microfluidic chip that he, of course, controls. On the y-axis, you see the rhythm of the endogenous oscillation that we measure. We started at 130, 140. This is where we have most data points. And then you see, as he starts to pump slightly slower, 140, 150, 160, the embryonic rhythm follows, and we start to slow down embryonic segmentation at least. So we measured the molecular oscillations. Paul measured the segmentation. He measured the different pathways. Everyone follows this rhythm that we provide from the outside. At least for the segmentation system, we are controlling developmental timing by using Arnold tongues. You see that it's finite. Above 190, there is no entrainment. We did see some occasional 2-to-1, 1-to-2 entrainments. I don't show them here, but I think that is possible. We should actually follow up on those. But what was clear is that it was much harder to speed up the system. We cannot entrain faster than 120. So it's already clear that for whatever reason, it seemed easier to slow down the system than to speed it up. So the question is then what to use this system for. And this brings me back to the initial motivation. Of course, the first question you will get is, does it matter to the embryo? Now, after many years, we are able to control the rhythm, and we would claim only the rhythm, even though you're, of course, never 100% sure, but at least we could not measure a change of intensity of these pathways. So we say we change only the rhythm, would it matter to the embryo? And this is what we then go back to a more complex system, a 3D system. The one I showed you before was, in fact, a 2D system that was easier to start with. But Simona in the lab now applied the same strategy to more complex structures. It's a 3D system that you see growing on the chip. Periodically, you will segment off, you will make the somites, and then you see the traveling waves. And you can ask what happens to the morphology. And to do that, she then quantified carefully the dynamics, elongation, and the segment width. And I only show you one snippet of her data. It's very much in progress and ongoing. But one finding I wanted to share with you that is rather recent, and that is the following. That is shown on the next slide. So if I show you here the results of the controls and of the entrained samples, the top are the controls. These are all the samples. I think these are 18. There are 60 here. And you see the average oscillation. The top one are controls that are not entrained. They oscillate at about 140 minutes. And here you see clearly the effect of entrainment. You can see that after 3, 4, 5 hours, the embryo switches gradually to this new rhythm and then oscillates at 170 minutes. So that was the change that we wanted to achieve. Does it matter? Is morphology changed? And here Simona shared with me this slide a couple of weeks ago. And what that shows is a quantification of the somites. So this is a somite here. And here you see somite size plotted for the controls in green. And she measures the somites as they form in the culture. So somite 0 was already there. And then 1, 2, 3, 4, 5, 6, and up to 12 somites were formed. And then she would measure the size of each somite. And you can see that in purple, this is the control. Actually, naturally, at least in this system, somite size tends to get bigger, smaller over time. So that's the comparison group. And then the green curve shows you the entrained samples. And that was very exciting for us because we see that after entrainment happens in this time window here, which is gray. So in this time window, the clock switches to 3 hours. It doesn't switch, it gets entrained to 3 hours. And then you see that the somite size changes. We also get a lot of defects that are more detailed. Somite size changes, as some prominent models would predict, but then somite size comes back to the normal size. We have to extend the time series, but it seems like this effect is transient. And that is very interesting to us. I will not go into much detail that is more specific to our field, but it is fair to say that the prominent model in the field that is in the textbooks would not predict that. It would predict that somite size gets bigger and stays bigger. Because remember, the period stays at 3 hours. We don't change the period back. But the phenotype is transient. And that requires new models. We were discussing that yesterday with Ertuğrul. There's other models that might be able to incorporate this sort of adaptation. It clearly hints that these oscillations and the dynamics, the change we introduce, some way feeds back to the spatial positioning of the boundaries in a dynamic fashion. And I think it's one example where now this top-down entrainment at least allows us to falsify the model, and now we will spend the next years of finding the next version of the model that can explain this finding here. And maybe for those interested in the details, we are now doing an optogenetic entrainment approach. The goal is to entrain not globally, as I showed you here, but to entrain locally along the axis of the embryo. So this is something we try to implement. Happy to talk about optogenetics with experts. This is challenging. Okay, so that was the first application of Arnold tongues. Of course, you can control the rhythm, what happens to morphology. But especially in this round, but I also do that when I talk to developmental biologists, I want to make a different point. I want to make the point that this perturbation approach can offer insight into something, in a way, more fundamental than that. It can offer insight into the very nature of the oscillatory system itself. And I want to explain what I mean. So if you go to, and I want to highlight that this, for many years, and more than a decade, has been a collaboration with Paul Francois, a theoretician in Montreal, and only because of that integration, this is possible. And again, this is exactly the spirit of this meeting here. Okay, so what we were discussing with Paul is the following. If you think of an oscillatory system, and depending to which audience you go, the first question you get is, how does the PRC of your oscillator look like? Okay, again, not really taught in development, but a PRC is a very important concept. The phase response curve of an oscillator, if you know that, it tells you really a lot about the nature of the oscillator. Phase response curve means that you plot the phase response on the y-axis as a function of the moment at which you apply the pulse, or the perturbation, I should say. Phase response curve. Again, I'm sure for many of you, this is very familiar. In our field, that would be less familiar. That's why I take a moment to explain it, just to be sure we are all on the same page. So for instance, if you hit the system at 12 o'clock, you see that the delta phi, the delta phase, is 0. If you hit the system at 12 o'clock, nothing will happen to its phase. If you hit the system with the same perturbation at 6 o'clock, you would see that you gain some phase. Essentially, you push the oscillator forward. If you do it at 12 o'clock, at 18 o'clock, you push it backward. Okay, so the response is a function of when the system is hit by the perturbation. And knowing the phase response curve is very telling, because in fact, the underlying bifurcation structure influences the phase response curve. And in turn, if you know the phase response curve, you can learn a lot about the coupling rules, the synchronization rules this system would show. It's that's why I call it a fundamental behavior of an oscillatory system. And then there's a second component to that I would like to introduce, and that has to do with phase response curves, but also with Arnold tongues. So I explained an Arnold tongue is a regime where you're one-to-one coupled, and now there's something even more, maybe even more interesting, and that is that within the Arnold tongue, you have a structure of different, we call that isophases. These are these lines here. And what these isophases mean is they actually show what kind of phase locking you have. Okay, so what I mean with that is that you could be one-to-one coupled peak to peak, so peak to peak, or you could be peak trough, peak trough, peak trough, peak trough. Both is one-to-one coupled, okay? You have the same frequency. In one case, your phase of entrainment would be peak. In the other case, your phase of entrainment would be trough. And that is extremely meaningful. I can sense that right now, because that actually underlying, for instance, chronotypes, okay? Or entrainment after jet lag. I'm wandering now across my Arnold tongue for sure. That's what I'm doing right now. I'm getting better every day. But once I reach my fixed point, I will have a chronotype. I don't know if you know your chronotype. I know my chronotype, and that's why I know where I am on this Arnold tongue. So depending on which line you are, this is where you sit in your Arnold tongue, okay? Phase of entrainment. And now, again, we can use theory, and there are books about this topic that make a very clear prediction. How to reach a particular isophase is determined by many things, of course, but one factor is the detuning. the mismatch between the two oscillators that we talked about a few slides ago. Okay, this is shown very beautifully here in a paper by the Herzl group in Berlin. They are mainly concerned with circadian rhythms. And on top you see the phase response curve, and on the bottom you see something related that is called the phase transition curve. These are entirely related representations of the same thing. You see here that the phase response curves told us that perturbation at 12 o'clock has no effect on the phase. Okay, if you look at the phase transition curve, we plot the phase at cycle 1. Actually, we plot the phase of cycle 2 over the phase of cycle 1. Okay, so you see this is the phase of cycle n, and this is the phase of cycle n plus 1. Again, you see that at 12, you end up at 12. There's no change of phase. If you hit at 6 o'clock, remember we gain a bit of a phase, you end up at 7 o'clock. Okay, so then the next cycle starts at 7 o'clock, and you gain a bit again. And you see what happens. For those of you familiar with this picture, you see what happens. You flow towards the right, and from here you flow towards the left. This is a fixed point. It's a stable fixed point. This is my chronotype. And now the theory predicts that the position of the fixed point on the diagonal is determined by the detuning. Okay, this is plotted here. Here, the detuning is 0. That means that the entrainment period is exactly the same as the endogenous period. Here on the left, there's a mismatch between these two periods. In fact, the endogenous period is a bit smaller. And so minus 24 hours, it's minus. So the internal clock is slightly faster. It creates a detuning, and that moves the fixed point to the right. And this makes some of you morning larks. Okay, and the opposite is true too. So here the mismatch is positive. You move the fixed point to the left, and you become a night owl. So the fixed point position on the diagonal is determined by detuning. And after discussing with the theoreticians, we thought, okay, then if that is a universal rule, let's test that in a mouse embryo. Is that possible to be seen, a chronotype, the equivalent, of course. Embryos have no chronotypes, but the equivalent of that, is that possible to be seen? And then we went back to the data where we shift the detuning across a range of periods, okay? And for that, Paul uses the same representation. We do the stroboscopic maps, so we plot cycle n plus 1 over cycle n. And essentially, we are looking for stabilization on the diagonal, okay? So here you see the first pulse, I think it's that one, second, third, fourth, fifth, sixth, and then at some point, it should get stabilized on the diagonal. This is the fixed point, okay? And at that point, it's a stable phase relationship, in this case, peak to peak. Okay, so this is what Paul did. Stroboscopic maps with mouse embryos. On the left, you see controls. The color is the time evolution of the experiment. The controls never converge to the diagonal, and that's, of course, expected. There's no entrainment happening. And on the right, we lump all experiments together, and you see the cluster around the diagonal. It's not so easy to see what happens. For that, it's easier to look at individual cases. So here you see 3 examples, embryo 1, 2, and 3, cycle 2, 3, 4, 5, 6, 7, 8, and then it stabilizes on the diagonal. The other embryo starts at a different location, stabilizes here, and this one is already there and stays where it is. Okay, so we can clearly see the embryos is converging to a fixed point, and we can determine its location on the diagonal. And then the key question was, is this position here dependent on the detuning that we can experimentally control? Okay, that's what theory would predict. Okay, so this is what happens. So here I show you 3 plots of 3 different detunings. 130 minutes, 150, 170. And in magenta, we show the fixed point of this batch of embryos. Okay, and the key point is that you see the location of this fixed point. This is pi over 2. And then you see that with a change of detuning, that location moves up the diagonal. And at 170, it's even further up. Okay, so that I thought was quite remarkable, that in this complicated system, in complex system, we see an effect on detuning on the phase of entrainment, as a theoretician would predict based on really pure theory. And here actually all of the experiments, I think these were more than 100 quantifications, and this fixed point moves very gradually up the diagonal to reach a new phase of entrainment, depending on the detuning. We thought that is remarkable, and here I show you again the phase of entrainment. This is the phase of entrainment plotted over the detuning range. And this is remarkable, I think depends on the audience you talk to. On one hand, when I show that to my colleagues in the developmental biology community, I want to point out that the same inhibitor that we provide, the same inhibitor brings about kind of a peak or a pulse to trough relationship. And initially people thought, well, that's because it is an inhibitor. But then the same inhibitor with a different dynamical regime entrains almost to the peak. Okay, so this is all dependent on the dynamical perturbation that we do. And then sometimes I get the question, well, is it still functioning as an inhibitor? And I said, yeah, of course, it can only function as an inhibitor. You cannot think about it in this perspective. You have to think about it as a dynamical system. And then you get a sense of why is this phase of entrainment sliding. It also means that we have now control over the rhythm and over the phase if we know the phase response curve carefully. The left side is interesting for theoreticians. I don't know who in the room is familiar with how these usually look, but that shape is very unusual. It would not match at all, for instance, the phase response curve that I showed you before. It simply cannot be a phase response curve associated with what I showed you before in the circadian clock field or something that we would consider close to a Hopf bifurcation. It doesn't match that representation at all. And it took actually very long time for Paul and the team to figure out what kind of representation would match that phase of entrainment behavior better from a theoretical point of view. And so we did with Paul, actually Paul did that with his team, an inverse problem. You take this plot and you try to figure out what kind of phase response curve that reflects and what theoretical model would fit this kind of representation. And this is what he found. So here I show you the experimental phase response curve that he calculated together with Victoria. It's very asymmetric. It's mostly negative. And this asymmetry is telling. So this asymmetry you would associate with a bifurcation type that is an infinite period bifurcation. For instance, a SNCC bifurcation, a saddle node on an invariant circle bifurcation. And to me, again, this is interesting because these bifurcations have very specific properties close to the bifurcation. For instance, the period diverges. This is not the case for Hopf bifurcations. There, the period does not diverge, but the amplitude diverges. So here we have evidence from the perturbation that we are close to an infinite period bifurcation. For me, that is real understanding, and that's why I put understanding with an asterisk. For others, again, I get the question, okay, but how does it work? Like, especially more in our communities, they would ask me, okay, but now what really have you learned by knowing that it's close to a SNCC, you see? And that's why I think we have to have a really active discussion of how do you define understanding? For me, knowing the class of bifurcations is a very important step forward. I got a lot of satisfaction knowing that. Now, of course, we have to turn it into something. We have to understand what is tuning this system close to the bifurcation. We think we are close to a SNCC. The modeling aspect showed that to mimic that, you have to think more of a relaxation type oscillator. We have seen many examples here at this conference. And I guess this would be more like class 1 excitability that would represent a SNCC and would have this asymmetric PRCs. And this again is interesting because from all the investigations in our field, for instance, looking at the time series or other modeling approaches, people have not thought of SNCCs. They always modeled it as an Hopf oscillator. All time series don't show you relaxation type oscillators. You see, and that's to me interesting. By looking at the system directly, you can only go so far. Here we don't look at it directly. We look at its response to perturbation to learn about the nature of the oscillator. And that's kind of the approach I want to follow and share with you. I really think of getting to these Arnold tongues that then Paul reconstructed. It could look similar to that. We could use phase response curve as a phenotype. I want to think of developing systems and think of how would the phase response curve look across the body axis, across differentiation, if you go from a stem cell to differentiation. How would PRC look like? And very importantly, how would the phase response curve look like if you go from a more systems level view, which I showed you today, down to single cells? Can we do a phase response curve of a single cell and see at what point across this spatial organization do these properties actually emerge? So this is what I would like to discuss with you and what we are going to try over the next years. Phase response curves are not easy at all, but I think that's a direction that offers a complementary view on what a phenotype could be. Okay. This was my key vignette I wanted to share with you. I wanted to show you a bit more the direction we are taking and the motivation to have an even stronger integration of theory. And that really has to do with also thinking of what is written on this poster here. How can biology inspire math or theory? And this example with the orangutans gave us several examples immediately. I think it happens every time you do that. On one hand, I said what we find is matching the orangutan theory like the textbook. That is true to some extent. But 2 or 3 findings actually don't match. And they're the interesting ones, of course. On one hand, we found that the system, while we entrain it, is learning the new rhythm at which we entrain it. In other words, if we entrain the rhythm to 3 hours and we stop entrainment, the system will keep going at 3 hours. And Paul and his team has found more evidence in the phase response curve that shows that the omega zero, that in all physical systems is not changing. In our system, omega zero is adjusting to the entrainment. There must be more feedbacks and slower feedbacks that feed back to the system itself and changes omega zero itself. Just think about that. I think that is very exciting. Paul and a student made a theoretical investigation of that. That is the paper I indicate there. I think that is a really interesting finding that pushes us to think more about what entrainment means in this context specifically. The second thing is that, and I don't show you the movies now, is that we found that even during entrainment, we see that this period gradient I was emphasizing so much re-emerges during entrainment. Okay, and that's particular because the entrainment is global. And that is special because cells seem to be able to couple to each other, they do, and to influence each other, even though the entrainment has a dominant effect at the beginning of the experiment. But over time, the cell-to-cell coupling seems to be very important, and cells evolve further so that the period gradient re-emerges. And this is something that is not understood at all, neither from the theory nor from a more kind of mechanism point of view. This will keep us thinking quite a bit over the next years. Okay, so as I said, I want to show you also the direction we are taking. The goal is to bring theory to more and more complex systems, and at the same time, I feel developmental biology and other fields are moving to more complexity for sure. And this is also what we are doing. We are very motivated to extend this view of internal rhythms to the external world. We know that we are in constant exchange with the world, and I was reading Claude Bernard a few months ago. He is known as the first physiologist, and he talked a lot about the milieu intérieur, about physiology and homeostasis. But he also has a full chapter on how the internal rhythms have to interact with external rhythms. For instance, day-night cycles or other dynamic changes in the environment. And that's what we want to consider now. How would the internal rhythms connect to the external world? This is also where this idea cycles on cycles comes from. And to do that, we started a new system 5-6 years ago, which is a very interesting fish system, Medaka. They come from mainly from Asia. They live in shallow paddies and rice ponds, very shallow water. They are known to tolerate a wide range of temperatures. But actually, the most interesting aspect for us, I mean, the main interesting aspect is this one. So on the left, you see the plot of temperatures that this outdoor facility would see. So the adults can withstand that. Breeding season is shorter. But the interesting aspect is the following. One and the same embryo, when it's laid and attached to the mom, would see day-night cycles of temperature. Because the water column is so shallow that the temperature fluctuates on this day from something like, I think it's 27 to 35. It's 5, 6, 7 degrees, depending on the season, that the temperature goes up, down, up, down. Now, this is an interesting problem for us. We have an internal system that oscillates at all scales, and on top, you have an environment that oscillates in temperature, which of course has massive effects on development. And we thought this is a system we want to study. So we brought Medaka into the lab. We developed the sensors so we can measure this dynamics. It's very difficult to see the oscillations. This is a knock-in reporter into a locus for transcription factor. It's a fusion protein, and you have to have sharp eyes to see the waves on the left panel, which is intensity. If you show it as phase waves, you can see the phase waves beautifully, one wave, one segment, one wave, one segment. Very striking oscillations. And of course, now in this system, we can modulate the temperature, either chronically. You see here 22, 27, 32. But most interestingly, we can go now up and down, and mimic the real world on the microscopes, and that's what we're doing. This embryonic clock, in difference to the circadian clock, is very temperature sensitive. I think most of you have expected that. There is no temperature compensation at the level of the clock or of the oscillator. The higher the temperature, the faster the clock. That is very clear. Interestingly, when we plot the dependency of the frequency on temperature, you see that here. We can plot the change, we can plot a Q10, and we were comparing that to other processes that happen at the same time in the embryo. How many minutes, Rich? Was that for me? It's good. 2 or 3 more minutes. And for instance, if you take the elongation of the embryo, we call that elongation velocity, that of course is also temperature dependent, and we measure its dependency too. And we see that these things don't really match. They both respond to temperature, everything responds to temperature, but quantitatively they don't really match. And we're trying to figure out how the system and different processes are integrated so that they're still coordinated and yield a morphology that is pretty much invariant. And I will skip over this slide and show the invariant morphology. So if you look at the morphology at the hatchling stage at least, we see that both the number of vertebrae or the size of the vertebra is pretty much invariant, independent of the temperature regime. So the question really is, and that's what we're tackling now, with all this change happening and this plasticity at the level of the clocks, how is information encoded in that system that has to respond to these dynamic changes, to then control morphology in a rather robust manner? And there's only one way, and that is to combine theory and experiments very tightly, and that's what we're doing, and I'm keen to discuss that more with you. We even want to go to the next level and increase diversity, so this is my outlook for development. It will be even more complex and even more diverse. We are sampling now Oryzias from different parts of Asia, and we're doing that because they all have different forms and shapes and timing of development. There's one species here called Minutilus. They're tiny, tiny, and then there are others that are big, fast and slow, and so this diversity is already there, and now we can bring it to the lab, measure clocks, measure the dynamics in 3D. Here I show you a cell cycle reporter that we made, and as in the previous movie, you see that also cell cycle is highly organized and spatially coordinated, and so that's the direction we want to take. The topics we want to tackle is this plasticity, but we're also thinking of topics of, we call it life cycle transitions. How do embryos pause development? These are future topics that I put here as a source for discussion tonight. And I want to finish by making the point again. I mean, at this meeting, I'm in the right company. There's only one way to approach this complexity, and that's by integration of theory. And at EMBL, we have launched an initiative that I would like you to know about. It's very synergistic with what is happening here. We called it Theory at EMBL. We couldn't really agree on theory plus something, like we just called it theory. It's very broad, and we are hiring new group leaders on the theory side. We have a sabbatical program. If you're interested, there's the code on the right. The sabbatical program is very important. Anyone interested to come to any of the EMBL sites in Heidelberg, in Rome, Barcelona, Cambridge, or Grenoble, you're really welcome to contact us. I think the goal is to mix different disciplines and start a discussion to then come up with a synergistic approach, just as you're discussing here. With that, I want to thank my team. It's a privilege to work with them, my collaborators, and you for your attention. Thank you.
Questions?
Thank you. It's an absolutely fascinating talk. So there is this interplay of these internal oscillations and then the stimulus that comes from the outside and the resonance between the two. So for me, the question is, when the internal oscillations respond to an external stimulus, In how far does this depend on their spatial arrangement and perhaps gaps in the spatial arrangement? You showed nicely at the beginning the system is 88 and the 89 then make a difference. I observed it was spherical in some way as a support, right?
Yes.
What happens if it's not simply connected, or if you put some holes strategically in places? What happens if it's a square grid? Would it work the same? So the question is, in how far does that system depend on geometry?
Yes, that's a perfect question, and something we have only started to look at because of the complexity of the setup. But we have two ways to do that. So on one hand, we use this in vitro system, where we can actually structure the surface. At least it's 2D, it's not 3D at this point, but we can impose geometries and then do the entrainment. So this is just the beginning, and we could also influence density to some extent. We have not really done that, but that is definitely something we have to do. On the other hand, what you mentioned is really important. We could think of gaps, at least gaps in terms of having no coupling partner.
Exactly.
And that we are trying to also combine with genetics, because we do know the molecules that are important for communication. And so now we want to combine this genetic approach and see what the effect of entrainment would then be. I think that's a very promising direction.
Pretty cool.
Yeah,
Thanks.
There we go. Hello. Thank you so much for a great talk. One moment where you were suggesting this idea that the phase response curve could be almost a phenotype, could be almost a characterizing factor, and sort of center your point of view there. That reminded me a lot of the work of Carmen Canavia in neuroscience, where she's doing phase response curves on different types of interneurons to sort of characterize their role then in the network. One thing, the way I understand her work, I don't know if there is anybody from her group here that could do the job better than me, but one thing I learned from their papers, from their work, is that there is a place where the strength of the signal, so that size of the epsilon, if you want, really can make a catastrophic change on whether you're getting an entrainment that then the cell can adapt to, then the cell can make its own, then the cell, to some degree, my intuition is understanding from what you're describing, that your system is sort of making this entrainment, leading into insightful changes that it takes with it, versus getting completely overridden by too strong of a forcing and sort of erasing what it's got available inside. Is there anything of that kind that you run into in your aspects of the research? Yeah, no, thank you for bringing this up, because indeed a lot of this motivation is from the neuroscience field. Reading all these fantastic books. Ichikevich is one of my favorite books, and that makes exactly this point, that to some extent a phase response curve can tell you about its potential function, integrators, resonators, and of course the strength of the entrainment is a very important point. The specific question you had about can you drive it into a catastrophic, we have not tested that or thought about that. Right now we are trying to stay in the weak perturbation regime, which is also very important to give enough time to come back to the limit cycle. But in principle, this is a motivation that we could also exploit, in the sense that if you have enough knowledge of the system, we could drive it into a particular state. That's right. Yes. I think that's a fascinating idea. We have to discuss that and think about how to implement that in this system. Yeah. But I think maybe a more general point is that I was wondering a lot about the neuroscience field has shown that this can be successful and can be done. And I feel like to some extent, the reason it happened there is because you had this readout, you have the action potentials, you have calcium. And I think that's what we're delivering now. That's a bit also what I wanted to say, that in developing systems, we can now offer this readout and this approach to dynamic gene expression, to protein dynamics, and be able to manipulate it. So in that sense, I think we could follow that path also now in expanded fields, and that could be maybe one way to think about it too. Yeah, thank you.
Hi. About one of the points you made in the latter half of your talk, you mentioned that the entrained frequency does stay, but in your experiment where you treated the embryo and trained it for, say, 3 cycles of somite growth, eventually it did go back. And I was wondering if that happened because you actually lost the periodicity. Maybe you could look at the notch activation waves to see if the periodicity went back to the intrinsic periodicity, or is it because the cellular proliferation pathways adapted and slowed down as a result, your somites went back to their smaller size, because those are very different pathways. That would explain that exact same data.
Yeah, I think that there are 2 points, and that I didn't explain it well enough. So in the situation where the somite size comes back, when it's transient, the period stays, right? So that's not a transient effect on the period level. Okay, so there we have a transient phenotype, even though the period, we keep entrainment, or so we don't stop entrainment. Okay, so in that transient nature, the period, I should plot the period stays at 3 hours. Okay, so that's one part, but the other part related also to the transient, or actually the adaptation of omega 0 of the natural period. I think that was also part of your question. We don't know what the mechanism is, and we also know that the effect is not everlasting. So if we entrain for 5-6 cycles, we stop entrainment, then the period is maintained for another 2, 3, 4 cycles, and then it comes back. This is also a bit the limitation of the system. You know, we don't observe for very, very long. I mean, in physical systems, you would observe for 1,000 periods. You know, we have 10, 12 if you're doing well. We don't know the mechanism of that, but now we're investigating what internal feedbacks must be in play to inform the core clock about this rhythm that it's experiencing. So that's something we are actively looking into. Yeah.
Hi, I wonder how much mechanistic information you can get by looking at the perturbations of the oscillators. Because, for instance, at a mechanistic level, usually they depend on things like kinases, putting phosphates on proteins, or proteolytic systems degrading our proteins. That requires a lot of ATP, a lot of energy. So by looking at the behavior of the oscillators, one could potentially discard certain networks or protein interactions that would produce one behavior or the other, right?
I'm not sure. I have to think about that, how you would discard that. What I do know is that it's a very complementary description also. For the wind pathway, we know very little. In fact, we do not know which mechanism produces the oscillation. Think about it. We don't even know the mechanism, and still we can control it.
But would there be a way to calculate how much energy does it take for a cell to do these oscillations?
Yeah, I think if you think of energy and also the metabolic background of that, that is something we quite actively look at. It's a bit tangential to your question, but maybe worth mentioning that you might know that also metabolism is rhythmic in many instances, and we are very keen to figure out if metabolism is one of the core drivers of what we're seeing here. So what we did in a recent work, we tried to entrain the signaling machinery with metabolites. And that works really well for particular metabolites in the glycolysis pathway around one particular enzymatic reaction, which is the phosphofructokinase reaction. It's not exactly energy, but at least it gave us an entry point into asking what is mechanistically or functionally coupled with the core clock. And to some, this inflow from metabolism is surprising, but we are pretty sure that entrainment helps us to differentiate its effect on the core clock. You see, because this is settled, but there's a difference of saying you really impact the variable, the phase, or if you impact more generally some parameter, see, like degradation of proteins and so on. And I think entrainment has a way to tell us that. At the abstract level, yet it will tell us functional dependencies. So we are trying to go in that direction. Yeah.